Today is the day we're going to be talking about reactive transport modeling. So I've been looking forward to this day. Hopefully you all have as well. This is the schedule for today. I'm going to kick it off with an introductory presentation. And then we have a tutorial session that will be led by Kuei Chen. And Hyun will give us another presentation later this morning on machine learning and artificial intelligence methods for metabolic modeling in reactive transport models. We have a little bit of a longer lunch break today because we have a Zoom conflict, and so we'll be breaking for a little over an hour, and then we'll come back in the afternoon with presentations by this group from Subsurface Insights, Roelof Versteeg and Rebecca Rubenstein, talking about some of the methods they have for incorporating microbial models into reactive transport. And then we'll have Michelle Newcomer giving a presentation on using some R tools for gathering and managing hydrologic data from the internet. So we're looking forward to a fun day with you. And again, please join us over on Discord for some chat and interaction. And with that, I'm going to go ahead and start up my presentation. So this morning, I'm going to be talking about incorporating microbial processes in reactive transport models. And so to just give us a little bit of a recap of where we've been this week and where we're going. This is a slide we presented early on in the course that gives an overview of the various elements of this course. And so you'll recall that on Monday, we spent a lot of time talking about metagenomes, metagenome assembly and binning and DRAM. And so we were kind of up in this area of the plot, and using some data from the WONDERS consortium, the worldwide consortium for collecting river from dynamic river systems. And then on Tuesday, we went into the other side of the WONDERS data, which is a lot about organic matter characterization using FTICR mass spectrometry and other methods, and analyses of those data and visualization of the results. And so we talked about a variety of tools down in this area. We also talked about how to use those data to derive models using lambda theory for reaction kinetics and stoichiometry. On Wednesday, yesterday, we really went into the deep end of metabolic modeling and how we use both metagenomic information as well as metabolomic information to construct reaction networks, describing the functionality of individual organisms, bins of organisms, or entire communities of organisms, and how to build those networks, incorporate metabolite information, and gap fill those networks. Today, what we're going to talk about is over here in this part of the diagram, where we take that information and actually begin to incorporate it into models that link hydrology, chemistry, and biology. And that takes us a step towards being able to apply these methods to understand and solve ecosystem scale problems. So I'm going to start by just addressing the question for those of you who may not be familiar at all with reactive transport models. What is a reactive transport model? And I'll give you a brief history of how microbial processes have been incorporated into subsurface reactive transport models over the years with some examples. And then I'll give you a description of early efforts that were made to link genome scale models with reactive transport models. And then finally conclude with current research and development areas on incorporating recent omics advances into RTMs. And a lot of this material is based on a recent paper that was published in the journal Elements by myself and Christoph Meile. Christoph is one of our instructors in the school, and he'll be presenting tomorrow morning. And I put a link to this paper over on the Discord site under the Reactive Transport Modeling channel. Definitely encourage you to take a look and check that paper out. It should be fairly accessible, written in the style for the Elements journal. So what is a reactive transport model? Well, this is actually the title of a paper by Carl Stiefel and others from 2005, which is a great reference if you want to learn about reactive transport modeling, and particularly how it can be used in research environments. And what they called reactive transport models are an essential tool and a new research approach for the earth sciences. And here's a link to that paper if you'd like to check that one out. Basically, this diagram is from that paper, and what they point out is that a reactive transport model couples representations, mathematical representations of hydrologic, chemical, and biological processes. Reactive transport models, or RTMs, are designed to quantitatively describe and predict the distribution of chemicals in time and space. And most RTMs have been traditionally applied in the subsurface environment, but certainly they can also be applied in rivers, in surface water bodies, in the atmosphere, and so forth. And so usually these chemical constituents are represented as being dissolved in water or some other fluid solvent. Could be air, it could be oil or gas, or maybe supercritical carbon dioxide. So they're solutes dissolved in a liquid phase or in a solid phase. They could be mineral phases, or they could be solutes sorbed to mineral surfaces, for example. And so when we talk about reactive transport, if we think about the two components of that, transport refers to the movement of these chemical substances through space, usually driven by the flow of the water or the other whatever fluid solvent they're dissolved in. And then the reactive part refers to chemical transformations that occur. And those occur through interactions with other solutes that might be in the system, as well as with mineral surfaces. And importantly for this particular summer school, interactions with biological agents. And so particularly bacteria, but also, of course, other microbes, fungi, and so forth. So here we have this diagram that from Carl Stiefel's paper showing an example of a system that could be modeled by reactive transport models. So here we have leachate coming out of a landfill. This would be probably rich with organic matter, and so it would undergo decomposition by microorganisms, and there would typically be set up this sequence of redox conditions spanning from very close to the landfill where all of the other electron donors have been depleted. You now have methanogenesis happening. And as you get a little farther away, you move into a sulfate reduction, iron reduction, denitrification, and ultimately far enough away where oxygen is still available, you could have aerobic respiration. Now, if we think about iron reduction zone and we zoom in on that zone, you might see, for example, microbes forming biofilms on the surface of mineral grains containing iron oxides. And as they're working to reduce those iron oxides to gain energy, they're releasing iron, dissolved iron, into the aqueous phase. They're also generating bicarbonate and other effects on pH, and the bicarbonate might then subsequently form carbonate minerals, including iron carbonates or calcium carbonate, calcite. As those minerals and biofilms grow, they in turn can change the properties of the porous medium such that you actually have a feedback onto the hydrology. So you might change, for example, the porosity or permeability of the medium and change the flow patterns. And so these coupling amongst these processes is very challenging to understand. And this is where reactive transport models can be very helpful to us in elucidating these complex interactions. So I'm going to talk a little bit more about transport first. What is underlying the processes of transport in a reactive transport model? Well, really, in most reactive transport models, there are three primary ways that things get transported in the system. The first is by what we call advection, which is simply movement that's driven by the flow of the solvent fluid. So if we look at this example of a pore scale medium, think of these whitish zones as being your solid soil grains, maybe sand grains. And in between them are pore spaces that water is flowing. And the blue dots here and the blue and green dots represent solute molecules dissolved in that water. So as we go forward in this simulation, what we see is these materials being advected through the porous medium, moving with the water flow, being carried as basically passive tracers in this particular example. And one thing we do see is that the advection process is spatially variable. So some areas where they're colored in green have higher velocities, some areas where they're colored in blue have lower velocities. And that leads to some important phenomena at the larger scale. In addition to advection, we can also have diffusion, molecular scale diffusion, which is movement or spreading of solutes that are driven by local gradients in solute concentration. So where the concentration is high, those solutes are going to tend to diffuse towards areas where the concentration is low. And typically that can be represented using Fick's law, which represents the rate of diffusion as a function of the concentration gradient. Now, dispersion is the third aspect that's often in our models, but dispersion is a little bit of a perhaps non-physical parameter, because what dispersion really represents is movement or spreading, apparent spreading of solutes that's driven by variations in advection or advective velocity that are smaller than the scales that we're resolving in our model. So in this case, if you think of this entire cube as maybe being one grid cell of our reactive transport model, where we're simulating things at a larger scale, we're not actually capturing all these advective variations. And so at the larger scale, what that looks like is it's spreading things out much like a diffusion process. And so in many reactive transport models, dispersion is also represented as a diffusion-like process using Fick's law. Although we've learned that in fact this kind of advective-dominated dispersion in many cases is not well represented by Fick's law, and what's often called in the literature anomalous dispersion is actually not anomalous at all. It's the norm. So you may see some other models put forward that represent this dispersion in different ways than as a diffusion-like process. But most reactive transport models will represent this process, these processes using these three kind of representations. And most often, diffusion is considered to be, molecular scale diffusion is considered to be small relative to physical or hydrodynamic dispersion, and so they're lumped together into a single term. Another example that kind of shows us how these things work together is this simulation where the black circles represent the solid grains and the red spaces represent the pore space. And we've injected in this system a little bit of solute into this blue zone. And as I move it forward in time, what you'll see is here we're going to allow diffusion to occur locally into the solid grains. And so in this simulation, now you see the diffusion is occurring into the grains as where the concentrations are high. And then once that solute pulse is passed, those can diffuse back out into the pore space, creating a very long tail on our breakthrough curve. And so these are the kinds of ways that advection, diffusion, and dispersion interact to move substances around in the porous media environment. Now, if we look at a reactive transport model, a typical model would be formulated looking something like this, where this term represents dispersion, which includes the effects of both molecular diffusion and physical or hydrodynamic dispersion. And that's incorporated into this dispersivity parameter or dispersion coefficient. C in this equation is the concentration of the solutes. Phi is the porosity of the porous medium. And then this term represents the advection, which is the movement with the velocity of the fluid flow, where U represents the velocity vector at the local point in space and time. And the way we generally then incorporate reactions into the advection dispersion equation is by adding a reaction term. And it's really this reaction term that we want to talk about a lot today. How do we represent that reaction term when we combine reactions with transport? So in particular, we're interested here in how do we incorporate microbial reactions. Now, there are many different kinds of reactions that can occur in environmental systems, and some of those are abiotic reactions. For example, interaction of solutes with mineral surfaces, sorption, desorption, surface complexation reactions. You can also have aqueous speciation reactions, where the various molecules are interacting in an abiotic context. But we also have microbes in our systems, and those microbes can gain energy by catalyzing reactions to move things closer towards equilibrium conditions. And they do that by breaking down or building complex molecules. And in so doing, they couple a variety of elemental cycles, including carbon, nitrogen, phosphorus, sulfur, iron, and many others. And by transferring these elements back and forth between or among various phases, solid, liquid, solute, and gaseous phases, they affect their physical transport and then therefore their spatial distribution. So as I think about how to incorporate microbial reactions, this table, which is from my paper with Christoph that I mentioned early on, we kind of described a series of ways that people have represented microbes in reactive transport models, starting with earlier representations of microbes as catalysts, then microbes as reactants, and more recently, microbes as active biological agents with some regulatory features. I'm not going to address the functional and trait-based model aspects here because Owen has already addressed that very nicely on his talk on Monday. But I'm going to talk a little bit about these first three and kind of walk you through with some examples. And the main example I'm going to use in a lot of these is the Rifle, Colorado uranium bioremediation site. You may have heard of the Rifle site. This is a picture of the Rifle site, and it is located in western Colorado along the Colorado River in the floodplain here. This is a former uranium mill tailings site, and so there was uranium mines, and where they milled the uranium and put the tailings from the milling process out into the environment, then the uranium subsequently leached and contaminated this aquifer. And so there have been a lot of studies here sponsored by the Department of Energy to look at how microbes can be used to help modify the movement of the uranium so that it doesn't go to the river. And so what you see down here is this little shed, and I'm going to zoom in on that shed. And what you see is an injection gallery of wells where the researchers here inject substances, in particular an electron donor, usually acetate, to stimulate microbial growth and activity. And then those microbes, when they're doing their thing, reduce uranium, which changes it from a relatively soluble oxidized state to a relatively insoluble mineral phase that it exists in the reduced state. There are many other reactions, of course, that happen in this context. And so we can have the entire redox ladder from aerobic respiration through denitrification, iron reduction, sulfate reduction, ultimately all the way down to various other reactions that occur in the process. Very often uranium is also reduced, as I mentioned. So these are some of the processes that need to be incorporated into our reactive transport model. So the first step of incorporating microbes was early on was treating microbes as catalysts for these reactions. And so an example of that is a paper by Wong and Van Capellen from 1996, where they studied marine sediments, and they observed that there is a typical profile of various electron acceptors in this system. So oxygen you would see very high at the surface of the sediment column, the interface with the water body, and then you'd see that depleted quickly, and nitrate also depleted fairly quickly, and then the buildup of reduced iron and manganese as these processes proceeded. And so they represented this in a reactive transport model as kind of indicated schematically here, where each of these processes was linked to the mineralization of organic matter, basically the oxidation of organic matter to carbon dioxide by microorganisms. However, in their model, they didn't explicitly represent the organisms. They represented the equations that they, or the reactions that they could catalyze by the various reactions in the model. But that assumes then that the biomass itself is in some sort of quasi-equilibrium. So there's no real growth or change in the volume of biomass in the system. And that the system is then constrained by the concentrations of the electron donor and acceptor themselves, and not by the composition or amount of biomass. Another example from the Rifle site, and actually most of the Rifle models do consider biomass explicitly, but I found one small example here from a paper by Yil and Fong et al., where basically they looked at the reduction of iron in phyllosilicates and clay minerals in the top equation. This is the stoichiometry, and in the bottom equation in iron oxides by iron reducing microbes. And what you see in these equations is that there is no biomass term. So the conversion of iron 3 to iron 2 by microbial iron reduction is represented using this stoichiometry, but the biomass is not explicitly represented here. And what they did acknowledge in their paper that this was an important consideration, and that would be included as new information became available in this system. Now, as we move forward in time, people began to recognize, including the rifle team, that the biomass itself was an important product and reactant in the system. And so now you can see that many of the models used at Rifle, example from Yabusaki et al. In the Journal of Contaminant Hydrology in 2007, you see now that this term appears in the stoichiometry, where the biomass of the iron reducers is explicitly represented in the stoichiometry, and the amount of biomass affects the rate of microbial iron reduction in the reaction term. And so this stoichiometry again represents the reduction of iron oxides to reduced iron 2, dissolved, and also now has this biomass term. And this is one of many terminal electron acceptors considered in this process. So when you look at the kinetics of the reaction in this model, you see that there are a number here, this N, number of electron acceptors, NEA, and those could vary from oxygen, nitrate, iron, sulfate, right down the ladder. And as each of those is sequentially consumed, this indicator variable chi is changed from 0 to 1. So initially it would be 1 for oxygen, and then as oxygen is depleted, it would go to 0, and it would become 1 for nitrate, and so forth. And so the way that these are often represented kinetically is in terms of a dual monode model, where the rate of the biological process depends on the concentration of the electron donor and the concentration of the terminal electron acceptor, parameterized by these half saturation constants. And the biomass yield in this model is based on an energetics representation from Rittman and McCarty. And the biomass itself is assumed to have a molecular formula shown here: 5 carbons, 7 hydrogens, 2 oxygens, and 1 nitrogen. And so that allows you stoichiometrically to balance your equations and represent the biomass in the system. And an example here is shown where they injected acetate into the system, monitored it at a number of monitoring wells shown in the various colored symbols, and then simulated it in the model with this dark blue curve. And you can see they injected acetate over a period of 123 days. That was the biostimulation period, and then monitored what happened in this system. And so if we look at the model results and compare them with the observations from Steve Yabusaki's paper, you can see here's a map of the injection gallery that I showed you in that one photo with the shed. And then you can see there are 3 rows of monitoring wells down gradient as the groundwater is flowing this direction. And so if we look at the results, we can compare results in each of these 3 rows of wells, with the data being the symbols and lines, and the model being the solid dark blue curve. And you can see that their model was able to quite well represent the movement of, in this case, a conservative tracer, bromide, and in this case, the electron donor that was injected, acetate. And here, the concentrations of sulfate as sulfate reduction occurred, and also associated with iron and sulfate reduction, the reduction of uranium and removal from solution into the mineral phase. Now, the next step from in this process, again, we started with microbes as passive catalysts, not explicitly represented. Then we moved to microbes as actual reactants, where they're involved directly in the stoichiometry and the reaction kinetics. Now we can consider the fact that, in fact, though, microbes are not simply another chemical constituent. They're actually self-regulating agents that can respond to their environment and regulate their chemical pathways depending on what their environment is. And this part of the story begins with a person named Derek Lovely, a professor at the University of Massachusetts in Amherst, who him and his team discovered an organism called Geobacter metallireducens. And this was back in the late 1980s. This organism was first isolated, I think, from sediments in the Potomac River, and was the first organism that was shown to respire carbon using metal, iron oxides, as terminal electron acceptors. And so it's interesting to me that, really only about 30 or so years ago, this idea of microbial iron reduction and respiration of something other than oxygen was a very novel concept. They also discovered another related strain called Geobacter sulfur reducens. And in terms of Geobacter sulfur reducens, Derek worked with Krishna Mahadevan to develop and curate by hand a very detailed genome scale model of this organism. So they mapped out its genome, they annotated that genome, identified proteins and their function, and developed a reaction network representing this. Now, at the time, Phil Long, who worked at PNL as a research scientist, was the PI of the rifle project. And Derek and his team were working on the rifle project because it turned out that Geobacter metalloreducens and sulfur-reducens were very important, were the dominant organisms at the rifle site responding to acetate injection and performing metal reduction. And so Phil saw this model that was developed by Derek and Krishna. They called their in silico genome scale model, and was intrigued by it and talked to me and said, we need to go talk to these guys. So we went and met with them in Massachusetts, and we developed a proposal to incorporate these new models into reactive transport simulations. And so this was really exciting. This led to the engagement with Yilin. Yilin was, and these are old pictures from back in that time. So Yilin at the time was, as you've already seen, very engaged in the research and the reactive transport modeling that was being done at Rifle. And so she was the one who implemented the genome scale model into the reactive transport models at Rifle, leading to a paper that we published in 2009 in Microbial Biotechnology, which was the first really to incorporate genome scale representation of microbial function into our reactive transport model. And we did that using constraint-based modeling based on what Hyun referred to as dynamic FBA, flux balance analysis, yesterday. And in that, basically, we do a genome scale characterization of the reaction pathways based on genomic sequencing and annotation to identify the metabolic network. And that metabolic network can then be represented as a stoichiometric matrix, which in turn can be subjected to flux constraints. In our case, we used flux constraints based on uptake of 3 key constituents: acetate, iron, and ammonium. And so these were measured in the laboratory and fitted to Michaelis-Menten type of models. And then those flux constraints were used to solve the dynamic flux balance analysis, depending on the local concentration of acetate, iron, and ammonium, using dynamic programming methods. And so the objective function used for those linear programming methods was optimization of biomass growth under the given conditions. And the way we coupled that to our reactive transport model was in this way. So if you consider a reactive transport model simulating flow and transport, it has a number of different grid cells. And at each time step in each grid cell, we know the concentration of these key electron donors, acceptors, and nutrients, respectively, acetate, iron, and nitrogen from ammonium. And so we would pass those as flux constraints to the genome scale model, solve the optimization problem to get the reaction fluxes, which then replace the reaction rates we would formerly have used from our dual monode kinetic model by the ones calculated directly from the genome scale model. And we use those rates to perform the next time step of simulation in the transport model, and then do the whole thing all over again. Now you can imagine that if you have a large complex transport model with many time steps, you're going through this process and running this in silico model many times if you did a direct coupling. And so the way we actually did that is we pre-computed a lookup table for a range of concentrations of acetate, iron, and ammonium. And then at each time step in the reactive transport model, we simply looked up a value that was close to the concentrations observed in that grid cell and used those rates from that lookup table. And I think you'll hear Hyun talk a little bit more about some advances of how we might do that better currently. So if we think about the outputs of that model and compare it with the previous model by Yabusaki et al., what we can see is that the genome scale model link is the dashed line, and the model by Yabusaki et al. Using the traditional monokinetic was the dark line. And what you see is both models perform pretty similarly in terms of representing the field observations. But the difference is, of course, now that the microbial reactive transport model based on the genome scale model actually has few, if any, tunable parameters. All of the parameters of the model are directly determined by the genome scale characterization of the metabolism of that system and optimization under the certain flux constraints of the, that are imposed. And so this is really nice in that this has moved us towards potentially a more predictive and less fitted sort of system. Now, a really interesting work that followed up, Yulin and Steve and Mike Wilkins and some other folks did some really interesting work by testing this model at Rifle against some other omics. These are observed indicators of gene expression, proteomics, basically measuring the proteins that are observed, which indicates which pathways are actually being expressed in the field by the microorganisms. And this graph over here from their paper shows how they compared the proteomics data, which are the blue bars, with the in silico model predictions under either limiting ammonium or excess ammonium conditions. And what you can see is that there's actually a lot of correspondence between the predicted pathways that were supposed to be active according to the genome scale model and the proteins that were expressed representing the activity of those pathways. And so they concluded in their paper that the most abundant proteins consistently match the highest metabolic reaction fluxes predicted by the genome scale model, which was encouraging. However, you can see there are also some differences here, and most of those differences were associated with nitrogen processes, nitrogen utilization and fixation. And strangely enough, the microorganisms out in the field were actually performing nitrogen fixation, which is a metabolically expensive process, even though there was abundant ammonium available, at least apparently. And so they used this information to alter the parameters of the in silico model to basically move it towards nitrogen fixation. But one of the interesting things is that given that the bulk observations of ammonium in the system, the activity of nitrogen fixing pathways would not have been anticipated. We would have assumed that they would just use ammonium rather than fixing nitrogen themselves. And so without having seen these proteins, we would not have. Was that my warning, Becky?
It's 9 o'clock.
Okay, great. Thank you. Okay, I'm just about getting to the end here. So these things indicated a key challenge in applying these models, and that's the challenge of scale. And there's two really indications here of scale issues or scale dependencies. And the first is that in our original paper in 2009, we actually had to modify the flux constraints by a factor of 10 in order to match the observed data. And that was because the predicted rates from the genome scale model were higher than what we were actually observing in the field. Okay, so although I said there was, that the parameters were specified, in fact, we did have to tune this one parameter in order to fix it. Now, the second indication is that the proteomics indicated that nitrogen fixation was significant, even when bulk ammonium was apparently not limiting in the field. So this leads us to believe that there are some issues that relate to fundamental processes of diffusion and mixing at the smallest scales. If you think about microorganisms sitting on the surface of a soil grain, perhaps reducing iron using acetate from the aqueous phase, what's going to happen is they're going to deplete that acetate locally, leading to a gradient in acetate from the center of the pore towards the grain surfaces. And in other words, the bulk concentration of acetate that we measure in the field or that we predict with our coarsely gridded model is much larger than the actual acetate concentration that the organisms are feeling or observing at the surface of the grain. And so what they're responding to in terms of their flux constraints is lower than what we think they should be responding to. And so therefore, their metabolic rates are going to be lower than we predict if we use the bulk concentrations in our predictive model. And the same is true with the nitrogen fixation. We could actually have ammonium depletion in areas where if we took a measurement from a well, we would see ammonium in the water, but the microbes themselves may not be feeling that ammonium or observing that ammonium, and therefore they're turning to nitrogen fixation. And so to look at this scale dependency, we did a numerical experiment using pore scale simulation. And this is work by Gazelle Tartakovsky et al. Published in 2013. What we did was we did the same process I showed earlier, but instead of the large scale reactive transport model, we had a pore scale flow and reactive transport model. And this was implemented using a method called smooth particle hydrodynamics, where the fluid flow is represented using particles. And so now for each particle, we have a set of concentrations of substrates that impose flux constraints, solve our model, get reaction fluxes and rates, and iterate on this process, again, using a lookup table approach. The results of this numerical experiment are shown here. We have two cases, a case shown in the first column with no flow, so diffusion only, and a second case where we impose flow from the top to the bottom and we inject acetate at the top. And what we did was we simulated the biomass growth, here shown in red, using by the reduction of iron, these green surfaces on the white grains. And this is coupled to the oxidation of acetate, which is the green solute in the pore spaces. And what you see in the case of diffusion only is that the acetate is being reduced preferentially in some areas, and in these areas there is actually a lot of more smaller grains and therefore more surface area, so that rate is larger there. And so now these zones have less acetate than the larger scale zones. And so if you look at the overall rate, if you would have predicted the overall rate from the average concentration, it's going to be lower in the actual system than it would be if we predicted it from the average concentration of the bulk system. And the same was true in the flow system, where we had an average concentration in bulk that was higher than the concentration being observed by the microbes at the grain surfaces. So my final slide here is that the future of microbial reactive transport models, and that's what we think, we've been talking a lot about this week, or moving towards this week. In 2009, we were limited to studying one model organism, Geobacter sulfur-reducens, and that model organism's metabolism was coupled to a reactive transport model through a pre-computed lookup table. When we thought about trying to incorporate complex microbial communities, this approach was really just not feasible. And of course, at the time, we really weren't able to develop microbial reaction networks for more than a few detailed organisms, because they were generally hand curated. Now, as we move to current day, 2020, we now have metagenomics, and we can use those metagenomics, as we've seen on days 1, 2, and 3, to develop genome scale reaction networks that represent the interactions of complex microbial communities. And as Hyun will talk about later in his talk today, we can use artificial intelligence and machine learning approaches to replace the lookup table to couple microbial metabolism and reactive transport models in a more efficient way. We can begin to integrate multiple omics data types, including metagenomics, metabolomics, proteomics, into our models. And we incorporate these into community modeling frameworks, as shown here in this schematic diagram, again from my paper with Christoph, where we use these omics data to inform genome and metabolic informed network models, set up reactive transport models, and perform parameter optimization by comparing with observed data using tools like, for example, Markov chain Monte Carlo methods, MCMC, and that process can iterate and then thereby improving our models over time. So I appreciate your attention here, and I hope there are some questions and discussion. I really look forward to having these basic concepts expanded on by our speakers throughout the course of the rest of the day.
Thanks, Tim. That was really great. I mean, that was such a nice walk through like the past 15 years on where we started, where we are now, and some of these accomplishments are still being forged. And this summer school is really kind of introducing a whole another generation of scientists to move this forward. So that's really exciting.
Well, I think that's why this school is particularly exciting to me, is that I'm seeing some of the ideas that, we developed early on, but we really couldn't do a lot with at the time.
Right.
But now they're coming to fruition because of the work of this community that is really enabling these things to become much more accessible and usable. So it's really super exciting to me.
It is. It is. So there are a couple of questions both in the chat and on Discord, and I think we have a few minutes to answer them. So if you don't mind.
Yeah.
So there's one question here from Ria Braun. What about microbial chemotaxis?
Yes, that's a great question. I love that question. So, Ria, you're totally right. Most of these models have treated the microbes as being a stationary attached phase. And so definitely when they're doing iron reduction, they are typically attached to the grain surface where the iron oxide minerals are. However, we have observed in the rifle site that during active biostimulation, there's a huge flush of aqueous phase bacteria, suspended bacteria or pelagic bacteria. And in fact, under those conditions, the geobacter do express flagella and they start swimming around. And we believe that they are chemotactic. So they do follow gradients in iron concentrations in the solute. Kind of think about it as like, well, I've used up my iron oxides locally, so I'm going to jump off, start swimming. Where am I going to go? I'm going to go where there's a bunch of other things going on, a bunch of other iron reduction happening, and that's going to probably be where there's a lot of iron 2 in the water. So they're chemotactic to the iron 2. We have done some work. I think probably Rishi Parashar is on this call. He might be on today. I know he's been on this week. He's done some really interesting work in this area. As well as other people. And so we've started to look at detailed models of microbial chemotaxis and microbial movement, including some studies, recent studies at EMSL. This past year, we had a postdoc come up and do some studies with microfluidics models looking at chemotactic bacteria. And so I can post a paper publication that we came up with on that recently. So yeah, that's a great question, very exciting. It's an area I think there's opportunity for definite improvement of these models by incorporating bacterial movements.
Here's another question from Britt Amerson. How many different microbes are you currently able to include in your active transport models?
Mm-hmm. Yeah, so I mean, back in 2009, we were happy if we could include 2 or 3. And of course, now with the metagenomics approach, we can really include everybody who's there. And it really depends on how you want to bend that. So we can treat the community as sort of a superorganism where we ignore the fact that there are cellular boundaries. And we look at the genomic potential of the entire community and represent that as if it were a single organism. And so that's one approach. But as Hyun, I think, has shown in some of his work, you can also then break that into compartmentalized systems. And those might represent, for example, your bins of your metagenomic system, where you bin them into different functional groups. And now you can represent each of those functional groups as kind of an organism per se. So generally speaking, we're not going to explicitly represent, say, 1,000 organisms in a community or 1,000,000 organisms, but we are going to bin them in some way. And it's really looking at how to appropriately bin them that I think is one of the cutting edge areas for research.
Yeah. So here's another one that really gets into the physical heterogeneity of the subsurface. And this is from Kare Anderson. What tools are there to incorporate the soil physical heterogeneity in reactive transport models? Is this usually done through differences in hydraulic conductivity, for example? Are there ways to represent anaerobic microsites, which could affect pore scale versus bulk reaction dynamics?
Yes, excellent question. So yes, there are a lot of different approaches for representing heterogeneity. And I think you're correct that permeability is usually the primary property of the porous medium that is represented in a heterogeneous manner. So there are a lot of geostatistical techniques that are the most common. If you look at stochastic models, reactive transport models, we can generate alternative simulations that represent the potential structure of the porous medium using spatial statistics method or geostatistics methods. And there's been a lot of research in that area. In fact, that's also sort of dear to my heart because that was kind of my thesis dissertation topic of study, was how to represent geologic heterogeneity in these systems. There's also, of course, chemical heterogeneity. And so looking at the different reaction rates and how they vary in space is also very important. And one way we can start to look at these things is by combining pore scale models models with larger scale models. And so as we look at the details of what's happening at the pore scale, we can gain understanding that helps us better parameterize the heterogeneous system at larger scales, properly accounting for pore scale heterogeneity. Because you're absolutely right. I mean, these microsites can be critically important to the performance of the system as a whole, but they're very difficult to represent explicitly. So again, the integration of pore scale and continuum scale reactive transport models is a really important area of research.
Yeah, that's really exciting. And I know you've been tremendously active in that area as well. And
Yeah.
Maybe that's next year's summer school.
It might be. Hopefully by then, yeah, we'll have made some significant advances in our EMSL multiscale modeling capabilities.
Absolutely.
So yeah, that would be a fun topic.
So there are a ton more questions, Tim. I think you're going to have some catching up to do on Discord, but I think we need to move on to our next speaker so that we don't run out of time at the end of the day.
Sounds great. Thank you very much.
Oh, Tim, thank you. It was such a great presentation.